Ok, quindi tutto quello che ho detto non l'ha sentito nessuno? No. Ok. Okay, I'm going to repeat myself. So, this is better because I had a dress rehearsal. Now then, I'm here to thank the people who are attending in person and our distinguished speakers who are bravely attending in person. Due to COVID restrictions, we have a very limited number of attendants in person. However, I would like to welcome and thank all the attendants who are following us online. This meeting, this workshop deals with membrane protein research. So we will talk not just of structures, but also of approaches for purifying, analyzing them, stabilizing them, a number of technologies which are at the heart of this beautiful yet recalcitrant biomolecules. And of course, we will have also sessions on the biology of these fundamental components of any living being, including ourselves. The workshop is part of the Prometeus network activities, and Prometeus is a consortium that joins very diverse expertise in order, this is what we told the European Union, but this is what we are going to do actually, to go from sequence to active molecules that would interact and utilize membrane proteins as targets. However, being scientists, what we really want to do is to produce knowledge and network diverse skills to achieve a synergy that at the moment we had a very, very difficult period, of course, because a RISE project is a project of people moving around. However, the consortium has been active and we are striving toward our goals. And of course, we also want to communicate about membrane proteins, in which we do truly believe. And across these 2 days, many distinguished scientists in the field will tell us what the contribution has been and will be in the field of membrane protein research. Now, I will finish now with my introduction, and I will give the floor to the chair of our first session, Paolo Mariani, also known as Professor Paolo Mariani, from the University Politecnica delle Marche. You may sit here, I think. Yeah. Maybe it's better. And then I will do this. And then you... Okay. Non lo scegliamo più. E così ti vedo. Ok, ci vedono tutti. Dopo resto qui. Poi dopo condividiamo il suo.
Okay, so Beatrice, thank you very much. And thank you to all the people here and in remote. I need to say that I am very happy to be finally in presence in a Congress, because it is a very long time, at least for me, that I wasn't able to participate, like all people, to any Congress, workshop, or conference. So I am very, very happy. And I would like to thank Beatrice very much for this effort in this time and to give us the opportunity to be here. So I think that we can... So then we start the first session of our workshop. And the first speaker will be Brian Kloss, which is here close to me. And he will speak about Brian Kloss from New York Structural Biology Center in New York. And he will speak about membrane protein structural genomics at COMPA, past and present, and I hope also future, to see what we will do tomorrow. So thank you very much.
should be able to make this full screen. How is full screen? If I want to make this full screen, everything's in Italian. If I want to make this PowerPoint.
Sei condivisione schermo? Ok. No.
Oh, this.
I think that this is a maximum. Schermo intero. Yeah? Yes.
Ah.
And you can close this one.
Oh. Okay, well, first, thank you for that introduction. And it's, in a way, it's appropriate, I guess, that I'm going first today, because so many structural biology projects actually do begin with us, literally, at Compa. So first, thank you to Beatrice for organizing this conference and for inviting me to share our work with all of you today. My name is Brian Kloss. I'm a scientist with the New York Structural Biology Center, and a member. Okay. Oh, can I? Is that okay? Okay. Is that better? Okay. So my name is Brian Kloss. I'm a scientist at the New York Structural Biology Center and a member of a group called COMPA, the Center on Membrane Protein Production and Analysis. And today I'll be sharing with you some work that we've done, sort of a historical perspective of our work, and possibly some ways of moving forward in the future. So when I speak of COMPA, I'm actually speaking of historically of two groups. The first is a group called NICOMS, the New York Consortium on Membrane Protein Structure, which existed for 10 years from 2005 to 2015. And this was a small-scale center funded as part of the NIH Protein Structure Initiative. And our goal was quite simple. Our task was simply to solve as many integral membrane protein structures as possible. In 2015, we underwent a bit of a rebranding and a renaming. The members remain the same. Many of the strategies remain the same, but the goals changed somewhat. Without going into them here, but this is the name of our group to this day. So for structural genomics, for those of you who aren't aware, structural genomics simply means using the natural diversity of sequence space that occurs through evolution and in nature to identify a protein target of any molecule related, say, to the one we're particularly interested in hopes of obtaining one suitable for structural functional studies. So no talk such as this would be complete without one of these introduction slides. So why is it we're interested in integral membrane proteins? Well, they make up approximately 30% of the entire genome. Genes encoding these proteins make up 30% of the genomes of all prokaryotic and eukaryotic organisms. A large number of drugs on the market today have as their targets integral membrane proteins. And yet these structures of these proteins of these class are vastly underrepresented compared to all the structures of all proteins known today. This is for many reasons. And most notably that they express very poorly in recombinant systems, and then they must be purified and extracted and purified in the presence of detergents, which tend to be expensive. One needs to identify the particular detergent in which the protein sample is happiest, and then you need to also find a detergent which won't interfere with downstream structural or functional studies. Now, on the right, I have a graph here of a slide taken from the website of Stephen White at UC Irvine, where he has been plotting the progress of membrane protein structures solved annually since the first integral membrane protein structure was solved. And as you can see, it has risen steadily over the years, even doubling in total over just the last 5 years alone. And again, there are many reasons for this, but certainly structural genomics approaches is one of them. So at COMPA, we can very briefly summarize our integral membrane protein pipeline, structural biology pipeline. We select, or our colleagues select a target in which they are interested in studying. We then identify through simple sequence analysis techniques like BLAST, although they're more complicated methods as well. of identifying all the related proteins related by sequence identity. In this case, we have a collection of prokaryotic reagent genomes, which I'll get into, or in the case of eukaryotic targets, most commonly these are either synthesized or purchased. We amplify these targets by PCR, and then we clone these targets into a variety of different expression vectors by ligation-independent cloning. And I'm not going to go into that method here, but suffice it to say, we think it's a far superior method of cloning as opposed to traditional enzyme ligation. These targets are then tested for expression at the small scale. In the case of prokaryotic expression screens, this is as little as 1 milliliter. And then we take promising targets and we distribute them to our colleagues for scale up and further study. Alternatively, we can scale up these targets in-house ourselves, even in a somewhat high throughput fashion. And then we can screen for detergent preference by size exclusion for any promising targets that will remain from the second round of screening. Again, these can be distributed to our colleagues at this point, or we can proceed with structural studies on our own in-house. So one of the strengths of NICOMS and still of COMPA was that we have amassed a significant collection of reagent genomes from various prokaryotes. Here is included basically our original list showing a diverse range of different bacteria, and includes several archaea, which in themselves include several extremophiles. This collection has continued to grow and now numbers almost 200 different genomes. And we have a particular emphasis, as Margarita knows, of an emphasis on the genomes from Mycobacterium, and we have nearly 4 dozen of them. So we've learned a lot of things along the way. And one of the most surprising things, which we still don't completely understand, but which we've tried to use to our advantage, is the fact that simply cloning the target of interest into a variety of different expression vectors, including vectors that otherwise look quite similar, in that they add either a small purification tag to the N-terminus or the C-terminus, can still give very different results when it comes to expression and purification. And below, I have two Venn diagrams of two separate sets of targets. In this case, two expression vectors that add a purification tag to the N-terminus, and two that add a tag to the C-terminus. And as you can see, these different vector types, which differ basically in their vector backbone, can give very different results. And as you add more and more vectors to your expression pipeline, the screening pipeline, you can significantly increase your rate of success of identifying potential targets for study. Now, like our genome collection, our collection of expression vectors has grown over the years as well. Until now, we have roughly 15 or more targets for vectors for expression in prokaryotic systems, and at least 10 for expression in eukaryotic systems, including insect and mammalian cells. And one of the kind of neat things about this is that, for example, here in it's an N-terminal set, we have these vectors can all accept the same PCR product for cloning. And so that it's quite simple just to take a single PCR product, insert it into multiple vectors, and go from there. Now, as you can imagine, if you have potentially hundreds of targets in different expression vectors, and then in different expression vectors, and I've not yet mentioned about using different expression hosts or different times and temperatures of induction for expression, these numbers of targets can grow significantly very quickly. And this then kind of necessitates that one has the ability to do high throughput techniques. In our case, we have liquid handling robots that can assemble all our PCR reactions, purify our PCR products, isolate mini-prep DNA, and even plate transform cells onto plates. We have the ability to grow small-scale cultures in large quantities simultaneously. In this case, using a high-speed shaker in which we can grow up to 1,000 different constructs simultaneously. We have the ability to do automated cell lysis using a 5-axis robotic arm onto which we've a sonicator probe. And lastly, we have the ability through a modified HPLC system to do automated detergent screening, or in the case of eukaryotic targets, fluorescent size exclusion chromatography. Moreover, we can then, in terms of scale-up expression, we have a fermenter which can grow 96 cultures simultaneously at what we call the mid-scale. They're held in a circulating water bath to maintain temperature, and they're fed a mixture of house air and pure oxygen. And you can see we can get extremely high cell densities as a result, with the cell mass acquired from a 50 to 60 ml culture in the system, roughly equivalent to that obtained from a half liter grown in a shake flask. In this case, we can then repeat the expression and purification of the targets that we first observed at the small scale, and verify that they express and still have enough protein left over to do some preliminary analysis of those targets. Here I show two targets which purified in near homogeneity after just a single affinity purification step, and each of these is then injected onto a size exclusion column to analyze its size exclusion profile. And in each case, we see a nice single monodispersed peak, which indicates a very well expressed and folded protein that can would now be considered suitable for downstream studies. Now, just in terms of a bit of bragging, so from our NICOMS years, this is the first 10 years of our group, we cloned and tested for expression over 16,000 different constructs. Roughly a third of these expressed at the small scale, and then through a lot of hard work and effort from our colleagues who were then given these targets, nearly 60 unique protein PDB structures have been resulted. Now, one of my favorite examples of the structural genomics approach is the case of the prokaryotic bestrophin protein. Bestrophin is an anion channel in humans, which is known, mutations of which are known to cause blindness. So a postdoc in Wayne Hendrickson's lab, Ting Yang, was interested in this class of protein, and she asked us to identify orthologs from amongst our prokaryotic species. There were very few, from what I recall, I think 3. And she managed to get, one of them expressed, and she then, through a lot of hard work and effort, solved the structure of this protein by X-ray crystallography. And I should say on the right, in this sequence comparison here, worth noting that these two proteins are only 14% identical at the amino acid sequence level. Yet she did solve the structure, and being an electrophysiologist by training, she was interested in seeing how this protein functioned, and more importantly, what happened in the case where there was a mutant protein. So she introduced into the prokaryotic protein a mutation that maps exactly to that commonly found in humans. And she finds that the function of the protein goes from a normal channel, which is almost always closed, to one that is nearly open. And the open probability is shown here below. So it gives a great, proves the value of this approach, and also gives some great insight into what might be going on in the human protein that leads to disease. And then, if you'll permit me, this is kind of a bit of structural eye candy, just a few galleries of many of the structures that have been solved by our colleagues, and many of which actually began their time at in the Compa labs. So since 2015, we've been known as Compa, and much of our hasn't changed, but there have been some changes, and most notably that we have now taken on smaller projects that involve eukaryotic targets rather than prokaryotic. And simultaneously, at the same time, other advances in the field have led to lead to our successes. This includes new detergents such as the neopentyl glycol detergents that have been shown to be very successful for GPCRs, and things like the synthetic digitonins, DDN, which are much more stable and less toxic to us. Lipid-filled nanodiscs, both of the traditional variety and circularized, that put the protein back into an environment that is much more physiologically relevant than a detergent micelle. Use of styrene maleic acid polymers that allow you to directly extract and purify the detergent directly from the lipid bilayer, and takes along with it closely held lipids that are believed to be important for proper protein folding and function, And last, but certainly not least, advances in cryo-electron microscopy, in particular, the development of direct detectors that have allowed so many structures to be solved at high resolution. And then just on the right, I have a small sample of some of the publications that have come out of this work. Includes quite a few eukaryotic targets, and I believe almost all of them have been solved by cryo-electron microscopy. So again, another thing we've kind of learned along the way, and which would be the basis of the remainder of my talk, and that is the idea of microgenomic expansion, which I'll get to in just a minute and define in just a minute. And it begins with a story of a sulfate transporter, a bacterial sulfate transporter called Substrate. This was part of our NICOMPS pipeline initially. What we did is we used the sequence of the E. coli protein here, which is highlighted in yellow, to then screen amongst our prokaryotic reagent genomes all the related sequences that we could identify amongst our collection. And these were then screened at the small scale and used to identify any promising targets. And from the screen, we found two, one from Idiomarina and one from Pseudomonas. And it turns out then that the structure from Idiomarina was solved in detergent by X-ray crystallography by a former student in Felipe Mancha's lab named Zara Assur. But for reasons not worth going into, she did want to obtain additional crystal structure. So I guess we could have gone and continued to screen additional genomes, but we thought, what if we use what we already know as a basis for leaping forward to try and find more promising targets? So the idea was that we could take these two promising targets from here and their sequences, and then find very closely related sequences to these two initial hits to identify more promising ones. Well, the Idiomarina genus is rather small, and we found only two. But Pseudomonas is enormous, and so we actually had to choose a few. So we chose 18, and these then were tested, cloned, and tested for expression. And here on the left, I have a cladogram, a very busy one of over 500 different CC sequences. And you can see the blue dots indicate the proteins, the genes that were tested in our initial screen. And then if you zoom in here on this kind of a highlighted part of the cladogram, we have the ones that were then tested for further study based on our approach. And they contain most of the Pseudomonas targets. And so what we did find, or what Zara found, is that neither of the Idiomarina targets expressed, but 16 of the 18 Pseudomonas targets did express, with several of them at extremely high levels. Several of these were then tested for their preference in detergent preference by size exclusion chromatography. We can see two examples here of a protein that are two proteins that would be considered quite content in a variety of different detergents. Several of them were crystallized, and here are 4 examples here, and then 2 ultimately had their structure determined by X-ray crystallography, and this is what led to this publication here, and would form the majority of Zara's thesis dissertation. So we thought, what if we could? Is this phenomenon that we're seeing? Is it potentially quantifiable? So what we did is we created 3 test sets for expression. One is what we call the pangenomic, or our initial genomic screen, our macrogenomic screen, if you will, of 36 targets. We then created a test set, or a positive test set out of our Pseudomonas targets. And then we created a third set, which we called our negative test set, which centered or surrounded the sequence of the E. coli protein, which didn't express very well or at all in our initial screen. We then expressed and purified at the small scale each of these targets in triplicate. And we included an internal control in each case for comparison, and then normalized their expression to that. And we found in these arbitrary units that the pangenomic branch has a mean expression level of 0.4. The E. coli branch does slightly worse, and the asterisk indicates that this is statistically significant. And the Pseudomonas branch, meanwhile, has expression levels that, on average, are nearly twice that of our initial test. So it seems that not only are we getting more things expressing, but we're also getting more things expressing at a higher level than from our original screen. So we then decided to take this one step further. So we selected the top 6 expressing targets from each set. These are indicated by the blue bars in this graph. And we then tested these for expression at the mid-scale. And as one would do in a typical purification strategy, you would typically purify it by affinity chromatography, in this case, nickel affinity, and you would then remove the tag by incubating it with a protease into which the recognition site was engineered into the expression construct, repassage that over nickel to remove the uncleaved protein, the protease itself, and the liberated tag. And to our surprise, what we found was that in the case of E. coli, we see 6 proteins, 5 of which express at decent levels, but not tremendous. And very little protein comes through in the flow through, suggesting that the protein is either precipitated or was unable to be cleaved. In the pangenomic branch, we found 1 out of the 6 made it through the step and gave a decent but not overwhelming size exclusion profile. Yet in the Pseudomonas branch, we found 5 of 6 that expressed very well and gave rather nice looking size exclusion profiles when screened in one detergent. What this kind of suggests is that these proteins, it maybe goes a step further, and it's not simply limited to whether this protein can be expressed or not, but whether this protein is properly folded, as would be suggested by its ability to be cleaved by the protease. And then, so here I provide a definition of microgenomic expansion, which is a revision of these standard structural genomics methods, whereby one, does your initial screen in a traditional sense, and then finds positive ones in which to really find that super sample, which is going to yield the best structure and be appropriate for functional studies as well. Now, I think, and what I'll get to at the very end of my talk, that this may be potentially very valuable for screening of targets of eukaryotic. So I wanted another example where I want to demonstrate another example, and that is the case of the mammalian protein STRA6. And STRA6 is a membrane protein which is involved in the transport of retinol bound to the retinol binding protein across the lipid bilayer from the outside to the inside of the cell. The structure of the xenopus protein, the zebrafish protein, was solved by Yunting Chen in Philippe Mancha's lab. But as you can see here from this alignment below, the human and the zebrafish proteins are still only 43% identical to one another. And while from our example for bestrophin, this certainly is valuable information, knowing the structure is valuable information, this difference in sequence identity could very well have implications on the final protein structure and certainly its function. So a mammalian, a human would be ideal, but a mammalian structure of the mammalian protein would be is highly desired. Now, it's already known that the human protein did not express, but the protein from the cow could be expressed but was highly unstable. So we decided to take that information and move forward with it by screening for related proteins, proteins related to the cow in sequence. So we then selected 6, which are shown here. And you can see that they're showing it pairwise. You can see that they're all still wildly different from the human protein, and were selected for having somewhere between 85% or greater identity to the cow. These then were used in a fluorescence size exclusion assay, or FSEC, whereby one fuses GFP to the protein, and then you can just collect a crude sample, inject it onto a column, and monitor its migration by fluorescence, and easily identify samples that are promising for scale-up expression. And from this, we found that there were 3: the cow, the cat, and the sheep, which looked promising by FSEC in this chromatogram here. And when those proteins were partially purified below, they still looked pretty good by given their size exclusion profile. The cat and the sheep proteins could be purified to homogeneity, and the sheep protein has subsequently been successfully incorporated into lipid-filled nanodiscs, and its structure has been solved by cryo-electron microscopy. So it looks like this might be a more broadly applicable approach. Which brings me to my last example, which is a purely theoretical one, the Duncan experiment, if you will. And That is of the protein called Z-iso. And Z-iso is an isomerase which catalyzes the reaction from this to this, and it's involved in the synthesis pathway of vitamin A in plants, which is, of course, important for the nutritional health of the animals that eat the plant, but also for the homeostasis of the plant itself. So these proteins are related to a bacterial protein called NNRU, of which there is no known function. The protein from corn, or ZMAs, has been studied extensively and is shown as indicated by this publication here. The protein could be very easily and well purified from E. coli. It was active. It was thoroughly studied for its catalytic activity, but a structure could not be determined. So I'm throwing this out there as another potential example of this approach. So in this cladogram on the left, it's obviously almost deliberately illegible. We identified over 500 targets from other plant species related to the corn, the Z-iso protein from corn. However, most of the targets were related to Z-iso almost entirely through the NNRU domain, and then varied considerably over the remainder of the protein. So couldn't really confidently be considered true orthologs of the corn Z-iso. This list was then kind of pared down even further by limiting the test set to those that shared were similar in length to the corn Z-iso protein and were indeed annotated as being Z-iso isomerase. And this then reduced the sample set to 149. But being eukaryotes and in plants, it's sort of a mixed bag. Many genes have introns and many do not. The Z-iso protein does indeed have introns in it. It would be very difficult for any lab, even one, a very wealthy one with a lot of automation available to them, to screen a number of targets of this size. So what we're proposing then is that one sort of selects a sampling of these, kind of equally dispersed throughout the list in sequence identity, to provide a kind of a broad coverage of the entire sequence space by testing only a minimal number of constructs. The sequences of those proteins are shown here in this alignment. You can see towards the C-terminal half of the protein, the NNRU domain containing part of the protein. They're very, quite a bit similar to one another. And then at the N-terminus of the protein, differ wildly from one another, and could potentially provide that the variability that would be would lead to better expression or folding and stability than the corn protein. When you compare each of these protein sequences pairwise, you can see that indeed you are covering a very large sequence space, that really none of these proteins are similar to one another at all. And that's that's the idea. Is perhaps then this could be used as a strategy going forward. This is a future part of my past, present, and future. Particularly for eukaryotic targets, and particularly for any laboratory, not just one already set up for high throughput expression. That could be kind of structural genomics for the masses. And so, just in conclusion, I'll just say that structural genomics approach strategies have been enormously successful. There's no question about that. But perhaps a modification approach of the approach, together with sample handling capabilities already available in most laboratories, perhaps only as much as a multi-channel pipetter to make your life easier, means that virtually anyone could take advantage of this strategy. Then I provide 3 examples where this approach could be used. The first, the need for different crystal forms. One, to identify a more stable ortholog, kind of to get past bottlenecks or hurdles that one encounters in their project. And lastly, and possibly a way for the same strategies to be used going forward, but for eukaryotic targets, and more importantly by any laboratory out there. And so lastly, I'd like to thank all the people with whom I've worked over the past 15 years, a large number of people from outside NICOMPS, NYSPC, and COMPA, my colleagues, current and past from NYSPC, And of course, the group with whom I work most closely, the COMPA Executive Committee, and in particular, our leader and principal investigator. I didn't realize that. Our leader and principal investigator, can't move that. Okay, Wayne Hendrickson, who's up there somewhere. And so with that, I'd like to end, and I'd be happy to take any questions. Thank you.
Thank you. Thank you very much. There is time for some questions. Anna?
I have a practical question. In your experience, have you ever solved the structure of the same membrane protein in detergents and in nanodiscs that is telling us what is better? I mean, there are differences if you solve the structure in these two different conditions.
Well, if first I must confess, I've never solved a protein structure in my life ever. so I can't speak personally, but yes, it's been done, and perhaps Filippo can answer. He can't answer. It's been done. I just don't recall what the conclusion was off the top of my head.
And you're asking between detergent and nanodiscs, for example. Most of the time that is, which is extremely reassuring, but the devil is in the detail. So for example, the structure of star 6 that Brian was showing, we have it in amphipol, which is a detergent mimic, and then a nanodisc. There are some substitutes. As you tell me, in ion channels, where the lateral pressure of the membrane has more impact, I'm not surprised that we see more relevant changes. So I think that in the first approximation, the structures are going to be the same. The lipid, it is a question of how the lipid environment interacts with the membrane protein. And in specific classes like ion channels, I'm not surprised that we are observing. My take-home message goes back to methods development with Brian. For the membrane protein, given single particle, if you can get it into a nanodisc, do a nanodisc. I've yet to solve one in a it's an I similar to Brian's, but in terms of solving it.
Okay, other questions? Some questions from remote. We can. No? Go.
Allora.
Sorry, we are controlling the volume. No?
Are there any questions from
Yeah.
participants online?
Yeah, it seems not.
Okay.
Okay. Okay, so we thank again Brian. And we go to the next speaker, which is Alexis Moreno from Calixar in France. And he will speak about native membrane proteins for drug discovery.
Questo? I do apologize, but in manager.
So hello everyone. Thank you for attending this meeting, and thanks to the Prometeus program and Sapienza University for giving me the opportunity to present the solutions that Calixar offers for the study of the native membrane proteins for drug discovery. So my name is Alexis Moreno. I have a PhD in biochemistry, and I'm a project manager at Calixar since 2020. Okay, so today I will speak about our platform, and I will focus my speech on one of our targets, which is the adenosine A2AR receptor, and I will present you all the work that has been accomplished with this protein. So a few words about the company. Calixar is based in Lyon, in France, and it was co-founded in 2011 by Emmanuel Dejean, our CEO, and Pierre Falson, who is our scientific advisor, and who also is a research director in the CNRS, an academic profile. And he's an expert in membrane proteins and especially ABC transporters. We are 17 people working on site, 1 CEO, 2 business managers, 6 project managers, 1 postdoc, 2 technicians, an office manager, and trainees. And we have a joint laboratory called Chem2Stab with the laboratory of Grégory Durand. It is a chemist lab, and it's located in Avignon University in France. Okay, so the company was created to answer the challenges to isolate membrane proteins in their native form. So Brian already talked about it, but membrane proteins represent about 50 to 60% of all therapeutic targets. It includes GPCR, the G protein coupled receptors, ion channels, transporters, and other families of membrane proteins. Among all the solved 3D structures available in the PDB, only 2 to 3% are structures of membrane proteins. And why? Because it's difficult to produce them and to isolate membrane protein in solution. And the conventional processes affect the native states of membrane targets. All of this leads on an impact on biodrugs, antibodies, vaccines, and diagnostic applications. So the solution that we offer for the isolation of native proteins is the development of proprietary molecules in collaboration with our chemistry lab. So these molecules act as detergents, but they are non-denaturing. This is really important for us. And we have 7 patent families of detergents that avoid the refolding of the proteins. We don't need any mutagenesis, truncation, or fusion. We improve the stability of the proteins thanks to these surfactants, and it allows us to purify proteins with high purity while maintaining their functional and structural integrity. Thanks. Okay, so we are offering 4 families of detergents that are available online. can purchase these detergents. So there is a Calixarin family, which gave the name to Calixar, to the company. The diglucose family, the lactobionamide family, and the tris-acrylamidomethane family. All of these conferring different amphiphilic properties to the detergent. And lately, we started to develop some SMA-like polymers that form nanodiscs. They are quite different from detergents, and I will speak about it later. So the Calixarin family, they are composed of an ionic Calixarin chrome that you can see here with carboxylic acid. So 3 polar head groups and an hydrophobic chain, which can be an alkyl chain like this, 8, or it can also be a cholesterol chain. We developed this molecule as a cholesterol derivative, such as the cholesterol hemisuccinate. And we also developed some other, not detergent, but stabilizing molecules, like this Calix gluc, with glucose grafted on the polar head. The diglucose family is made of diglucose, polar head. It's a non-ionic detergent that allows small micelles formation and mild detergent properties. The lactobionamide family are non-ionic too, and they come into 2 flavors. They can be either with an hydrogenated alkyl chain or a fluorinated chain. And the TAC family for tris-acrylamidomethane family are made of a bulky polytris polar head linked to hydrogenated or fluorinated chains that allows tunable detergency properties. So a few words about SMA. So SMA are styrene and maleic anhydride acid copolymers. They allow the extraction directly from the membrane. So we can extract directly the proteins from the membranes by keeping the native membrane environment. So this is a good thing because we keep the native conformation of the protein. It is compatible with cryo-EM, and they are very stable over time at 4 degrees. However, there is a poor reproducibility of the purity of commercially available batches, and the solubilization rate is sometimes not that good. And also they are sensitive to acidic pH, and they are sensitive to cationic ions, which is a big problem when you work with biological targets. Also, it gives low IMAG yields. There is a glitch here, sorry. So here, just to show an example of the SMAs that we are currently developing. So we improved the synthesis of the SMA to get rid of the impurity that we can find in the commercial samples. So here we solubilized membranes from bacteria or insect cells for 2 hours at 4 degrees. And then we centrifuge the samples to recover the solubilized fractions and the pellet, which is the unsolubilized fraction. We did it on 3 targets, protein targets, BMRA, ACRB, and A2A. And here you can see the SMA 2000, which is the commercial SMA on Western blots. So it gives these big smears at low molecular weights. So it extracts nicely the proteins as our, but there is this smear. Okay, so we don't have anymore this smeary bands with our new SMA, and they extract as much, even more sometimes, as the commercial one. And here you can see on the second Western blot, it's the same problem with ACRB, and for A2AR, which has a low molecular weight posed a big problem because we couldn't run the SMA 2000 on the same Western blot as the others because it will just hide the protein totally. Okay. So we developed detergents and we use them for the purification of membrane proteins that we also offer on our website. So we have some GPCRs available, A2AR and the melatonin receptor NTR1. They are involved in neural protection in order to develop new treatments for cancer, cardiovascular diseases, insomnia, pain, depression, Alzheimer, and Parkinson. We also offer transporters, ACRB and BMRA, involved in multi-drug resistance. So we study them for the improvement and validation of novel molecules as therapeutic antibodies or new antibiotics. And we are working on several other targets. Another GPCR, the cholinergic receptor M4, involved in motor control and Parkinson's disease. The transporter MRP4, which is a multi-drug transporter upregulated in endometriosis and dysregulated in several cancers like neuroblastoma. We also work on the chikungunya spike protein for the development of a vaccine. The CD20 receptor, which is clinical validation targets of monoclonal antibodies in the treatment of lymphomas, leukemias, and autoimmune disease. And finally, the ion channel KCC2, which is the powerful therapeutic target to find treatments against pain and epilepsy disorder. Okay, so now I will speak more about A2A. So the adenosine A2A receptor is located in the platelets, in the brain, cardiovascular system, and immune system in the T cells. Its role, it's an adenosine receptor, of course, but it's also a dopamine glutamate receptor. It's involved in vasodilatation, inflammation protector, tumor growth regulator. The pathologies associated with this protein are acute encephalopathy, Parkinson's disease, insomnia, pain, depression, and hypertension. So we produce the full-length wild-type protein only with affinity tags for the purification of the protein, separated by a TEV cleavage site in case we want to get rid of the tags and only have the native protein. We express A2A in SF9 insect cells with the baculovirus infection. And we perform some solubilization assays. So here, this is a negative control. So this is before the treatment. Here, there is no treatment. And after 2 hours of solubilization, you cannot find anything solubilized, and it's all in the pellet. Here you have the positive control with SDS, everything is solubilized, but it's not in a native active form. Here it's DDM-CHS, which is commonly used for GPCR extraction and purification. But as you can see, it is not really effective because you have like half of the protein remaining in the pellet and half is soluble. And here it's one of our proprietary detergents, the Calixar. And as you can see, we have a very good solubilization of A2A. So we chose to solubilize the protein with a mix of Calixar and some DDM-CHS, just because after the solubilization process, we purify the protein only with DDM-CHS. So here you can see the purification of the protein by IMAC. immobilized metal affinity chromatography. We have a purity of over 90%, and we can produce up to 5 milligram ml of protein. And we provide it in a PBS buffer complemented with DMCHS. And as you can see on this is a stain-free gel, and this is a Western blot with an anti-8-way antibody. As you can see, we can still see some dimer. It's naturally a dimer, even if it's an SDS peak. And there is some C-terminal clipped parts of the protein here that you always have. Okay, so then we checked the quality of our protein. Here we use the radio binding assay and we showed that our purified A2A was able to bind both an agonist, the CDS compound, and an antagonist of A2A, so the Zm compound. This means our protein is probably active, and it's in a native form. We also performed some chronostability assays just by letting the sample on the bench for 7 days at room temperature, and then we analyzed with the steric exclusion chromatography whether we had aggregation or not. And as you can see here, we have a nice peak, and there is no aggregation, which would be at the beginning of the elution chromatograph. Okay, we also evaluated the thermostability of the protein. So here you can see in light blue the protein extracted and purified with DDM-CHS only, with melting temperature of about 45 degrees. Here we also evaluated the mutant A2AR*2 protein. So this is a mutated and truncated protein that has been designed for improving its thermostability. So indeed, it improves the stability a lot, around 55 degrees, but the protein is not native. It is a truncated protein. And in dark blue, you can see our protein extracted and purified with Calixarine and then DDM-CHS, which has also a great improvement in thermostability compared to the DDM-CHS alone, and which is a full-length, non-truncated native protein. Okay, so after purifying these samples, we send it to different partners, and at Institut Cochin, Université de Paris, Université de Strasbourg, and Molomix, to study the activity of the protein. So Stdnmr experiments were performed. Here we checked that we had a binding of known agonist-antagonist molecules. So here we could detect the binding of caffeine, which is a natural ligand of A2AR. We also detected the binding of the agonist CGS, and we were also able to see a displacement when we added caffeine after added Zm, which is the antagonist of the protein. Thanks to this work, we can confirm that we have the same binding pocket for both molecules. Then we screened small molecule fragments with the same method, Stdnmr. And here we evaluated the CAMP response in cellulo, because GPCR, when they are active, they are responsible for the production of CAMP. And we could identify 10 fragments that displayed either strong or weak binding, and among them, 6 behaved as antagonist of our native A2AR. So a second work was performed with affinity selection mass spectrometry by Edelris. And they screened about 200,000 compounds on our protein. And we could identify 2 compounds that behaved as antagonists of A2A. So here you can see the agonistic effect. This is the control agonist, another one. And here are the molecules we didn't find any agonist. But we did find 2 antagonists, which here, the 5 and 9, which reduce, not totally, but they reduce the level of CAMP produced by A2AR. Okay, so we studied these 2 molecules more precisely. And as you can see here, for the compound 5, we have a full antagonist. But for the compound 9, it's only a partial antagonist of A2AR. We also performed some allosteric modulation assays by adding CGS agonist and adding also different concentration of the compounds. And we could identify compound 5 as an inverse agonist negative allosteric modulator, whereas compound 9 is only a negative allosteric modulator of A2AR. Okay, so finally, hydrogen deuterium exchange mass spectrometry was also performed for an epitope mapping. So here we could identify the peptides containing important residues for orthosteric binding. And there was the possibility to evaluate either if the binding was orthosteric or allosteric. And here we have the possibility to combine these results with a native mass and NMR that were previously done. We also performed some mice immunization trials using A2AR as an antigen. So 3 mice were immunized either with our protein or with the mutant protein STAR2. We performed an ELISA after that, and we could see that our native A2AR here exhibits a higher immunogenicity than the mutant protein regarding the native A2AR found in the membranes. Okay. So just to conclude with our services. So we also offer our services for the production of native membrane proteins, starting from molecular biology to the expression, extraction and stabilization, purification and quality control of the protein. And we also offer the scale-up with up to 25 liters of mammalian cells that we can produce at the same time, and proteins in the milligram scale. We have different expression systems to do that. So mammalian cells, egg or CHO, yeast, insect, bacteria, and we offer the protein purification either with micelles of detergents, with SMA-like polymers in nanodisk or in proteolipase. For the analysis, we are recently equipped with a flow cytometer for the expression of GFP constructs. We can perform fluorescent thermophoresis and Western blot. We also assess the thermostability of the protein with fluorescent thermophoresis also. CPM thermal shift assay, Western blot, cryostability, and we can analyze the homogeneity of the protein with size exclusion chromatography. We have 2 ACTA pure purification system and 1 ACTA purifier. We can perform native page, DLS. And finally, we also check the activity of the protein with ligand binding, as I showed you, with radio binding or fluorescent polarization or thermophoresis. What are the outcomes? So we published a small application notes in collaboration with nanoimaging services, where we purified ACRB, this is a trimer, in Calixarine only, without any DDM, DDM-CHS. It was only Calixarine. So the purified protein is way more stable in Calixarine than when it is purified with DDM, as we showed here. It is active. It could bind one of its ligands, the rhodamine 6G. And we were happy because we could also solve the structure of ACRB in Calixarine. And the structure is quite similar to the structures already available, but this was the proof that our detergents can extract, purify native membrane proteins. They are active, and we can have the structure by cryo-EM. Okay, so we published also a lot of work, but some more work on CD20 in complex therapeutic antibodies, and we have some work about KCC2 and BAC3. And that's it. If you want more information, you can come to me or visit Calixar website, calixar.com. And thank you all for your attention.
Thank you. Thank you very much for the presentation. Some questions, please. No, not yet. Dan, but there is a question from here. Okay, yes.
Okay, so these are experiments that are running right now, but there is no correlation between more protein extracted and more activity. Like with SMA, the protein are in their native environment, so you can be quite sure that it will be active, but you have a very low yield. With detergents, okay, so sometimes you extract a lot of proteins, but not in an active form. So it is really protein dependent. Here for A2A, for example, our detergent works really well. The protein is active, so maybe it's not interesting to go to SMA, but it's not the case for all the proteins. I think it's very protein dependent. You have to check for each protein. Okay, so our construction is not supposed to be cleaved. There is some natural cleavage, unfortunately, as you can see on the gels, but our is just the full length proteins. So yeah, I think.
Please.
Terrarians, detergents. Now you hear me. Okay. So what is making this special? I see and I notice that nicely actually stabilizes some of your targets, but what is making this so special? Can maybe you improve this by doing something else? You know, going in another direction, taking into consideration what you have improved now in these molecules. I'm not sure to understand your question. You mean some other experiment that can prove the stability, or? So I noticed that in comparison to DDM, your Calixar detergents improve quite a bit the stability of the protein. So that means that the molecule that you have designed has some very important properties that is making this better than DDM. So I was wondering, what are those properties? Maybe you have studied this already, okay, and whether you can improve that. You can go beyond that. Okay, so why it works better? We are not sure, but once we find a good molecular scheme, let's say, we just produce we produce a lot of different Calixar. We screen them for different targets, and we try to improve with drug design, but sometimes it doesn't work as we want. So for now, it just okay. Let's try a bunch of different molecules synthesis, and let's see what works. Thank you.
Okay, Anna or Filippo, you have a question? No, Anna, please. And then.
So if I understood correctly, they work like SMA. They remove the protein from the membrane, right?
Well, the SMA-like polymers that I showed, yes, but the other surfactants, they act as detergents, so they form micelles and we get rid of.
But the Calix, the one of the A2R receptor,
yeah, so the Calix are just detergents, so we don't have any nanodisk. I didn't show anything about it, and we are still working on it, but we have some of our detergents that also form nanodisk.
Okay.
Still remain.
My second question is how expensive are they compared to the other detergents?
You can go on our website and see. Big purchase. They are a little more expensive because we are a small company and we don't have big facilities to produce them.
Okay.
But, yeah, I forgot to say that we also offer a screening kit. So this one is the most affordable one, I think.
Okay, last question.
So have you an idea on the mechanism by which detergents solubilize protein? I mean, if they form micelles in which proteins are included, or they mix with phospholipids and facilitate the removal of the protein?
Okay, so they form micelles. We studied this actually, and just like regular detergents, they form micelles, and we don't have any lipids. There is a total delipidation around the protein.
Filippo?
Did I see it right in your A2A example? You show it's a mixed system with some DDM-CHS.
Yeah.
So I have two issues, I have two questions on that. One, the comparison with the DDM-CHS is at 0.5%. Mm-hmm. I worry a little bit that you're, based on the ratio of the membranes to the detergent, that you might be not soluble. I mean, we all know that DDM-CHS works while you're getting a 50-50. I don't think that's a fair comparison to poor old DDM. But what is interesting is, do you need DDM-CHS in combo, or can you go solo? Okay. And do you have any idea of what's going on at the micelle size?
Yeah, so okay, so to clarify a bit, so when we did the purification in DDM-CHS only, we extract with 0.5% DDM. And after, during the purification process, we lower the concentration to 0.05%. When we do it with Calixarine, we extract with 1% Calixarines complemented with 0.05% DDM-CHS. And we are not sure of what we have during the purification process because we don't add any more Calixarine because it tends to concentrate. And we suppose that we still have some Calixarines with DDM-CHS around the protein. And we are not sure about this, but we can see some differences even if we don't add any Calixarine after during the purification process. And yeah, so we don't really know. And we didn't actually perform the whole purification with only Calixar. It was performed at the time because CHS was shown to be important for GPCR, and in the literature everyone was doing DDM-CHS, and we didn't do pure Calixarine. We should, I think. But it was done a while ago.
Suggestions was that you should maybe talk to Brian. It would be lovely to have to take 1,000 membrane proteins and do a microscale test of the efficiency of this detergent. And with the central membrane protein production analysis, they could do this quite readily on a very small scale to see efficiency of solubilization. It would be nice to see it,
Yeah.
How does it perform on the 1,000, 5,000 targets?
And I invite everyone that want to stabilize their targets, contact us, because we don't have this bank of.
No, that's why the two of you should talk.
Yeah. Okay, thanks.
Daniele, a short question, please.
Mm-hmm. Mm. Yeah, we don't have any data on, let's say, heterodimers, because A2A can also form heterodimers, dopamine receptors. We don't have this data. We have to develop a test, I would say, to assess the oligomerization of the protein in detergents. We have to do protein-protein interaction assays, but yeah, we still have to do it.
Okay, I think that we can stop. Thank you again, Alexis.
Thank you very much.
We go to the next speaker, which is Bernadette Byrne from Imperial College. The title of the presentation is New Approaches to Working with Membrane Proteins. I think that you need to open the screen. Yeah.
you. Okay, thank you very much, Paolo. Thank you very much to Beatrice and to all of you for coming to this meeting, both face-to-face and remotely. So for myself, this is my first foray out of the UK for over 2 years, and it's particularly lovely to be in this beautiful city on this particular occasion. So it's really nice, actually. I think my talk follows on quite nicely from both Alexis's and Brian's. Here I'm going to talk very specifically about some of the proteins that we've worked with and some of the issues that have arisen in some of the questions we've also tried to explore with some of our proteins. Okay, so I'm going to talk about, the talk is split into sort of 3 parts. The first is looking at protein engineering, which we know can be extremely powerful for stabilizing proteins in a form that's suitable for structural analysis. I will touch on novel detergents as well, as you've heard from both Brian and Alexis how important these are. Our research on this is focused on the novel detergents that started with the MNG molecule, published originally in 2010, but through a subsequent collaboration with the Technical University of Hanyang Technical University in South Korea. We've developed many other versions of those and characterized those with our particular target proteins closer here. Okay. And then finally, I'll talk a little bit about a novel lipid screen that we developed because, you know, we all know now how important lipids are for membrane protein structural function. It's really only in the last sort of few years that we've been able to explore these effectively. Okay, so I'm going to start, of course, with the protein engineering aspects. Okay, so before I go into that, though, I'll just talk about the actual molecules that we're most interested in. These are secondary active transporters, which are responsible for transporting molecules from one side of the membrane to the other. And they do this in an ion-dependent manner. So they harness the energy that's generated through the formation of an ion gradient across the membrane to drive the uphill movement of a substrate against its concentration gradient. Hopefully you can see that little animation there. This is how it works. It's a coupled process. So these type of secondary active transporters tend to be two-domain, with substrate either binding to a site that's formed by both domains or only to one domain. Okay. So there are various mechanisms by which these proteins function, and these are summarized in this very nice diagram that was included in a review paper by Olga Budka and David Drew that was published a few years ago. I won't go into the specific details of this too much, but I'll just sort of draw your attention to, to this elevator mechanism here, because this is the mechanism by which our particular transporters function. This involves the substrate binding slowly to a single of the two domains. And then what happens is you get this movement of one of the domains against the other, and this pulls the substrate across the membrane. And I'll come back a little bit to that, the importance of that in a moment. Okay, so here I'm just showing you a membrane protein structure that we solved a few years ago now. This is a protein called UAPA. It's a uric acid xanthine transporter from Aspergillus nigricans. And here you can just see this in the dimeric form. So here we've got one of the protomers shown in a ribbon representation, and the second shown in space filling representation. So what was absolutely crucial to obtaining this particular structure was a single point mutation. And this single point mutation was substituting this glycine here at position 411 for a valine. And you can see the two different structures here. So this is the wild type form of the protein, and this is the mutant. And what you can see here, so here we've got one of the domains of the protein shown in red and one domain shown in blue. And this particular residue sits at the interface between these two domains. This red domain here is a substrate binding domain. And it's this that slides up and down as the protein undergoes its elevator transport mechanism, pulling the substrate from one side of the membrane to the other. Now, the introduction of the valine here, projecting out into the space between the two domains, we think prevents this action taking place because you've just got additional bulk projecting into that site, and this stops the substrate binding domain moving against the dimerization domain. So clearly this is important because not only did this result in non-functional protein, but it did result in much, much greater stability compared to the wild type form of the protein. And ultimately, of course, gave us the high resolution structure. So then we asked, because we were working on other structurally related proteins, could we transfer this stabilizing mutation into another protein? Okay. And I should say that the protein that we chose to do this is not in the same family of solute carriers as UAPA. So we decided to use a protein called BOR1, which is a boron transport from Arabidopsis thaliana. And this is actually an SLC4 family member. UAPA is an SLC23 family member. But the two families share very, very close structural homology, and I'll show you that in a moment. So here we've got the structure that was obtained from Bob Stroud's group that was published in 2016. This shows you the two protomers of the protein. So again, like UAPA, this is a dimer. So here I'm showing you a superposition of UAPA in the dark blue, dark pink or red color with the AT-BOR1 in the pale blue, pale pink. So you can see without even knowing the RMSD values that these two proteins do indeed superimpose very, very well. So what we were most interested in is this region of the molecule here. So in the red color here, you can see the valine that we introduced into the UAPA. And what we were trying to do is we were trying to identify which residue was the equivalent one in AT-BOR1. Okay, and I'm just showing you this view of the protein from both the inside and outside of the membrane. Now, it was a little bit difficult to precisely identify which was the correct amino acid residue to mutate. And this is probably due to conformational differences in the two structures. So UAPA is inward facing and AT-BOR1 is inward occluded. But we were able to identify two possibles. So one was proline 362 and the second was methionine 363. So we generated some mutants of both of these sites individually. And what you can see here is fluorescent size exclusion chromatography. So this technique that Brian talked about earlier. We express our protein as a GFP fusion in Saccharomyces cerevisiae. So maybe if we just start at the bottom panels first, these are the P362 mutants. And clearly what we're trying to do here is we're trying to add bulk into this particular site. So we substituted the proline with either a valine or a leucine. So if you look first, this is the FSEC profile that was obtained of protein unheated, so at 4 degrees C. And then, so you can see here that the wild type protein, which is shown in black, gives a reasonable recovery. But both of these mutants actually give slightly better recovery at 4 degrees, so unheated. But if we heat this protein at 46 degrees C, which is a little bit above the TM for AT-BOR1, which in our hands at least was about 43 degrees C, we get absolutely no recovery of monodispersed protein. So this indicates that these mutants are not any more stable than the wild type. It could be less stable, but from this data, we can't really tell. But if we turn our attention to the M363 mutants here. Here we substituted with either a phenylalanine or a tyrosine. You can see again, we got very similar results to the P362 mutants at 4 degrees. But importantly, when we heat it at 46 degrees C, we see a complete loss of wild type recovery, as you'd expect, given the previous results. But we see substantial recovery of the two mutants, indicating that these are both more stable than the wild type and the P362 mutant. So in order to assess what effects this has had on the function of the protein, we carried out functional complementation analysis using a delta BOR1P knockout yeast strain. So what happens in this yeast strain is it will grow quite happily in the absence of boron. But if you introduce boron into the growth media, what happens is this accumulates inside the cell to toxic levels and the cells don't grow anymore. Only when you introduce a functional Bohr transporter, and that doesn't have to be the yeast one, it can be the 80BOR1 homolog, do you see growth of this knockout strain. So here you can see the growth of this particular strain that was expressing the wild type form of the 80BOR1. And here you can see that both our mutants, the M363F and Y, gave much poorer overall function, indicating that our protein has stabilized, probably as a result of reduced conformational flexibility and thus reduced function of the protein. So we purified both of these mutants. I'm just showing you the M363F version here, just for clarity. So here's the wild type protein, which really uses a very nice sec profile. The M363F also gave a very nice sec profile. Here it looks like the actual recovery is a little bit lower, but I think that's just batch-to-batch variation. I think overall we got at least as much protein back this mutant typically as we would for the wild type. And then here we've got the SS page analysis for the two different proteins. So here you can see that for the wild type protein, we almost always saw quite a sort of, it wasn't a particularly attractive SS page gel. Always got these two bands that we think are the dimeric form, and an additional band here that's almost certainly the monomer form. And what we see with the thermostabilized mutant is that we see much less of this heterogeneity in the sample. We got crystals of both forms of these. This M363F mutant, the crystals did diffract slightly better than the wild type, but not sufficient for us to sort of write home about. So clearly more engineering probably required. Okay, so why is it that one of the mutants works and not the other? So what we think here, the reason we think the M363F mutants work is shown here really. So here we've got in panel A, we've got the 80BOR1 wild type structure. So here in pink is the P362, here's M363. If we now mutate the P362 residue to the leucine, you can see that this, while it substantially increases the bulk in this region of the protein, the overall bulk from the two residues combined is not significantly different compared to the wild type. The methionine residue sticks out really quite far into that gap between the two domains. This M363F substitution, however, does project much more, much further into the region between the two domains. And we think it's that's reducing the conformational flexibility of the protein. Again, it's doing what I'd suggested with the UAPA. It's trapping that substrate binding domain and preventing the conformational rearrangements required for transport. Okay, so now I'll talk a little bit about the novel detergent work that we've done, again in the context of a specific protein that we've worked on. So you've heard mention already of the MNGs, and these were crucial for this particular project. So here I'm talking about a protein called OS43, which is a RISE homologue of ATBOR1, the protein that I just talked about. So here what you can see is the sec profile that we obtained for this particular protein in DDM and DTT. So just to sort of say here that this is a C-terminally truncated version of this particular protein. We tried working with the wild type form of the molecule, but this proved to be nigh on impossible. And it was only once we removed a big chunk of the C-terminal domain were we able to recover any protein that we could work with. The DTT here was proved to be crucial for reducing the levels of aggregation of the protein during purification. So here what you can see is the sec profile for this particular construct in the presence of DDM and DTT. And okay, overall it doesn't look too bad. We've got a bit of an aggregation peak here, and this peak here probably corresponds to some free GFP that's sort of carried over after our cleavage event. Now in panel B here, you can see the STS page analysis of the individual sec fractions. And it's this band here that we think corresponds to this particular protein. So although this didn't look too bad, when we also did the in-gel fluorescence analysis of these individual fractions, we saw that there was an additional band that runs a little bit higher that's lighting up even after this purification event, indicating that we haven't got full cleavage of the GFP. And so although this looks okay, it's really not as homogeneous as you'd expect in the sec profile. So one thing that's standard in our lab is also to do some thermal denaturation analysis in, as Brian and Alexis have referred to, a panel of detergents. And here we use, of course, DDM. It's even with our years of experience of screening novel detergents, we always have a look at DDM in the first instance. So I'm sorry, I'm really having trouble with this mouse. So here we've got DDM in the black. You can see it's not quite the worst detergent. The protein doesn't unfold quite as badly in DDM as it does in non-R maltoside, which is, of course, the worst detergent. It's what you'd expect here. It's shown in the pink, but it doesn't look great. Okay, and after 2 hours of heating at 40 degrees C, you really don't have much folded protein left in solution. What was notable here was that all of the MNGs and even the octyl glucoside version of that, OGNG, gave much, much better thermal properties, conferred greater thermal properties on the protein. So one of our, I mean, we did many, many, many things with this protein, but I'm just going to talk to you about those, the screening work that we did with those detergents in particular, because clearly it was this that made a massive difference. So here we've got this particular construct purified both in LMNG and DMNG. And both of these gave very nice sec profiles. And you can see here we get this very nice STS page pattern for both. We do see an additional band here for the LMNG purified protein, but this doesn't seem to be due to the presence of uncleaved fusion protein. Now, the sharp-eyed amongst you might notice that there's quite a substantial difference in the retention volume of the protein in the two different detergents. So here in LMNG, we have a retention volume of about 12 mils, and it's substantially less than that in DMNG. So, and actually this is probably reflected a little in the STS page analysis here. So what's happening here is that the detergent is determining the oligomeric status of the protein that we're isolating. So in LMNG, the protein is isolating as a dimer, which is the physiological form of the protein as far as we're aware, whereas in DMNG, this is isolating as a monomer. Okay? Even though it looks beautiful and it is extremely stable in both forms in our hands, it turned out, oh, sorry, yes, let me just discuss this. So we did some negative stain EM analysis on the 2 different samples, and this confirmed what we thought. So here in DMNG, you get these small, almost sort of spherical-like particles indicative of the monomeric protein, whereas in LMNG, we see these almost like sausage-shaped particles here that are much bigger and that are indicative of the dimer together with the detergent micelle. And the dimer proved to be absolutely essential for further structural analysis, and we were able to get a low-resolution structure for this RISE boron transporter in the presence of LMNG. And the presence of the dimer here was absolutely critical for doing that. And of course, it is the most important because it is, we think, the physiological form of the protein. Okay, so now I'll move on to talk about this lipid screen work that we've done. So we know, as I mentioned right at the beginning of the talk, that it's becoming increasingly clear that lipids are crucial for membrane protein structure and function. I'll just give you some nice examples here from the literature. So this is the structure of the betaine transporter, BET-P, and it's bacterial. I think it's E. coli, but I can't remember off the top of my head. But here you can see the different protomers of this trimeric protein. And crucial for forming this particular oligomer are a number of PG molecules that drive the formation of that particular oligomeric state. And then another example is given down here. I think this is a PE molecule as identified from the structure, and this has a role in modulating the function of this ligand-gated ion channel. And then here, as many of you will be aware already, without me telling you, it's really, really well known that lipids have major roles in GPCR structure and function. And this is just a diagram that we put together for a review we published last year. So here we've got cholesterol, which is crucial, we know, for oligomerization, function, and stability of GPCRs. GM3, which is a glycolipid, plays a role in allosteric modulation of GPCRs. And then, of course, phosphatidyl'inositol bisphosphate is becoming increasingly well known as having crucial roles in protein-protein interaction, particularly between GPCRs and beta-arrestins and proteins, but also has roles in activity and stability of these proteins. But of course, as we're all aware, the process by which almost all membrane protein purification occurs through the use of lipids removes most, if not all, of the lipids associated with a given membrane protein. Detergents are designed to disrupt the membrane and break it down into small pieces and effectively replace the lipid. Lipids are also lost during isolation stages. So certainly in my lab, we really try and limit the number of purification steps we do with any particular protein, because we know that an additional step can often result in catastrophic lipid loss. You can relipidate, of course. You can add in specific lipids once you've purified your protein. And there are some very nice examples where this has been done. And this is a nice structure from Rob McKinnon's lab of the Shaker potassium channel, where addition of phosphotylglycerol, phosphatylethanolamine, and phosphatidylcholine had crucial roles in obtaining this beautiful structure here. Another example is Circa, which crystallized in the presence of PC. If you're not sure what lipids might work for your individual protein, then you can use commercially available additive screens. So Hampton and so on make these, and you can add them in very, very small volumes to your crystallization files. And these can work really very nicely. But the amounts that you've got and the process that you're doing means that you get very little information back on the precise role of individual lipids, and it's certainly not suitable for nanodisk preparation or cryo-EM analysis, or for other biophysical techniques such as native mass spec and HDX So my former PhD student, Cristina Sacchetti, decided to go about designing a cost-effective lipid screen as part of her PhD. And this formed part of a collaborative project with ourselves at Imperial, together with University of Leeds and our commercial partners, Molecular Dimensions and Anadrolix. So we started by putting together a database of lipids known to have roles in structure and function. And we used the Lipid Maps database for this, the Protein Data Bank, and literature search. And also we knew quite a bit as well in our own lab. And then Cristina and the other students put together a business case for the company, and they went about designing a cost-effective lipid screen. So this comes in a 96 well plate format with 32 different conditions. In order to try and make this cost-effective, we did exclude expensive lipids. So phosphatidylinositols, which we know are important for a lot of membrane proteins, unfortunately are also extremely expensive. So we left these out, at least in this pilot scale. Where appropriate, we did use different versions of the same lipid, where there were examples of both in the literature that had contributed to stability or crystallization or any other characteristic of a specific membrane protein. But we did try to cover as much lipid space as possible. I'll just give you here a couple of example conditions on top of the ones I've already mentioned. So this is some work from Carol Robinson's group in the University of Oxford, looking at the role that specific lipids have in oligomerization of transport proteins. And this shows you the native mass spec data that was obtained for the leucine transporter. And her analysis, or her group's analysis, showed that cardiolipin was absolutely crucial for lutein dimer formation. Similar work from our own group looked at the yeast BOR1 transporter and showed that phosphatidylserine was crucial for dimer formation of this particular transport protein. So these conditions were also, of course, all included in our screen. So just quickly to go through the production process, the lipids were brought in by the company in chloroform. They were dispensed onto a 96 well plate, and then the chloroform was evaporated with nitrogen. We then solubilized the lipids with the particular buffer that we're interested in that's suitable for our particular protein. And then we add our detergent solubilized protein. And then in our case, we use nano-DSF to look at the stability of the individual proteins in each of the lipid conditions. So we had 3 test proteins. So the adenosine 2A receptor, which now needs absolutely no introduction, but just to highlight here that this construct used here was slightly different than the one that Alexis has talked about. This has this Brill molecule inserted into the 3rd intracellular loop. This was expressed in insect cells. Our UAPA protein, this thermostabilized molecule, expressed in Saccharomyces cerevisiae, and then a paraphosphatase from Thermatoga maritima, again expressed in Saccharomyces. And here's just to show you the nice quality of protein we've got for all of these particular proteins. A2A never looks that pretty on a gel. You always see at least 2 bands. Assume these are monomeric and dimeric forms of the protein. And because of the glycosylation, you always get this slightly fuzzy band. So that's why this one doesn't look as good on the gel as the other 2, but the protein prep was very high quality. Okay, so I'll show you the results for all 3 proteins using the same format. So what you're seeing here is the delta TM that was obtained for each of the proteins in each of the lipid conditions compared to protein in the presence of no additional lipid. Okay? And each lipid group are color coded. So here we've got the PEs shown in this, well, you can see that sort of a cream color. PCs are in blue, and so on. So we'll start by looking at the adenosine X-ray receptor. This is extremely well known for having crucial interactions with cholesterol. And normally it's purified in the presence of CHS, the more soluble salt of cholesterol. But here, because we wanted to assess the effects of specific lipids on our protein, we isolated the protein in the absence of CHS. So the first thing to note is that in our particular screen, we did see a very positive effect of CHS on the stability of the protein. Okay, so this is nice because it validates our approaches in terms of the usefulness of the screen. We also identified something that had never been identified before as possibly important or playing a role in the structure and function stability of this particular protein, and that was phosphatidic acid. Just as another positive control here, so sorry, I should say about this phosphatidic acid, this is certainly something, you know, worth following up on to see exactly what role this particular lipid has in the structure of the protein and function of the protein. Here's another positive control here. This is the substrate ZM241385. One other thing to mention here is that as part of the screen, we also included these two monoacyl glycerols that are used for lipidic cubic phase crystallization. And what we found here was that we got a very strong negative effect on the stability of the protein in this particular assay. So either this isn't the correct way to assess this, the interaction between this protein and these molecules, or these genuinely are having a destabilizing. Okay, so now we've got the same kind of data for UAPA. So one thing to say is that from our previous work, we knew that PI and PE have a very important role in dimer formation of this particular protein. But interestingly, the addition of more PE didn't seem to have any positive effect on the stability of this protein. And we think this is to do with the fact that UAPA co-purifies with PE and PI already. So adding additional PE doesn't seem to have any specific effect. What we did see was that in this case, the PS versions that we looked at did substantially stabilize the protein, not significantly, but certainly something to follow up on. Okay? We've never picked up PS before in any of our other analyses in terms of co-purification. So if PS is playing a role in the structure, function, modulation of this protein, it's definitely something that's lost during the purification process. And this is something we're very keen to explore through nanodisk reconstitution and native mass spec analysis, as well as possibly cryo-EM. So there were some other sort of quite marked negative results. So the phosphatidic acid molecules really destabilize this particular protein. Cardiolipin 2 really wasn't happy in there. And as we saw with the A2A protein, we also got this destabilization in the presence of the monoacyl glycerols. Then our final example is this paraphosphatase. Now, this was a protein that we had no idea whether this had any lipid dependency. Interestingly, this particular molecule seemed to be stable in virtually all of the lipids that were added. So all of them increase the stability of this protein. There are a couple of examples where you saw less of an effect, but overall, this seems to be a kind of non-specific effect. That lipid possibly partitioning into the detergent micelle, therefore stabilizing the protein. A sort of notable, again, were the MACs. So these are clearly not so good for our screen. Okay, so we know that we've got to work a bit more on optimizing this. So yes, as I was saying, we've got to optimize our screen conditions. We need to work out the long-term stability of the screen. That's not something that we've had a chance to do yet, under different conditions. So at 4 degrees, at 20 degrees, and so on, and frozen as well. We'd like to look at a larger number and variety of membrane proteins, so with more or less TM domains, different functions, different sources, and so on. And nano-DSF is a fantastic technique, but we don't actually have it at our institution, as do many other institutions also don't have it. So we're really quite keen to also explore this usefulness of this with other stability analysis tools like the CPM-based assay, which is something we use much more frequently in our laboratory. We also want to use the screen as a means or a resource in order to facilitate crystallization trials and/or nanodisk reconstitution in a high throughput manner. But of course, this is all dependent on funding. We've been turned down once and we're currently under consideration again. So to summarize everything that I've said today, it is possible to transfer stabilizing mutations between structurally related membrane transporters. And the reason I wanted to talk to you about this today was that for many laboratories, resources are limited. And so to start to do like a large scale mutagenesis or engineering approach, even alanine scanning, it's just not possible if you have 600 amino acids within your protein. So any information that can help guide you is useful. So have a look at what structures are out there, see which ones are similar to yours. If people have used stabilizing mutations, try them. It's relatively quick, relatively cheap to do. Detergents, of course, we all know this. I'm not telling you anything new here, that can have profound effects on the purity and oligomeric state of the membrane transporter. One thing that has come out of the discussions from both Brian and Alexis's talks are the fact that we often don't know what we're doing when we're adding something into our protein. We just go, oh, we've got 17 detergents, let's just stick it through. And I think this has been the way we've worked with membrane proteins for a really, really long time. And what I'd meant to include, but didn't, was that we got the proofs yesterday for a very extensive review of the 25 or 30 or so novel detergents that we've worked on with our collaborators at Hanyang, which are now starting to allow us to see why specific molecules behave in the way that they do with respect to individual membrane proteins. And this is particularly related to detergent-detergent interactions and detergent-protein interactions. So I'll send that paper to Beatrice because it's quite a long read. But it really does give you a lot of information about all of the novel detergents that are out there within the context of SMAs and so on as well. But it also provides a template for rational decisions about which detergents to use for your given protein. And then finally, I presented this novel lipid screen that we developed, which has been very interestingly allowed us to identify some new lipids that we didn't know anything about that might have roles in the structure and function of both a GPCR and a secondary active transporter. And just finally to acknowledge, so almost all of the work that I've presented today was done by Cristina Sacchetti. Savas did the work on the OS-BOR3 molecule. Nikki helped out with some of the mutagenesis on AT1. Our collaborators at Warwick and the University of Leeds, and then finally our funders. And I really do have to particularly thank RAMP, which is the consortium that funded Christina, and BBSLC. And thank you very much for your attention.
Thank you very much. Thank you very much for the interesting presentation. And there is some time for some questions. Okay, please.
So thank you very much. Very interesting. So I also tried to add MAGs to a protein that actually had been crystallized in lipidic cubic phases. So obviously it was a good one, but as an additive, as a lipid additive, made it precipitate. So just to add on to that. And then maybe a suggestion that as a cheap assay for stability, it is quite convenient to use GFP. It's not as a fine assay, but just looking at, upon eating, how much protein has survived the heating, is heat centrifuge and either run a gel or measure the fluorescence.
Yeah, so we do use those kinds of things. The issue here, of course, is that what we're working with when we're doing our lipid screen is the final product. So we express many of our proteins with GFP, but it's removed during the GFP, during the purification. And so we want to know what lipids are going to positively influence the final product, which has no GFP on. So yes, at lipid screening stage, it's got to be something that's non-fluorescent based, at least not GFP. It's not dependent on GFP. Thank you.
Next, please.
I had a small question concerning this stabilizing mutation or mutation. Did you try to look whether some of this stabilization was actually related to the fact that the side chain of the new amino acid, the mutation, is actually filling up a gap, a void, an empty space inside?
Yeah, it's a good question. So in UAPA, so the protein is it's in the inward-facing conformation, and the G11V projects into the translocation channel, and so it's not actually filling a void because it's actually in the structure. This translocation channel is packed with detergent molecules, but so it's not filling it. There is a space within the protein that can accommodate it easily. But we think what's happening is it just prevents this movement that's crucial. So by having this sort of extension into that translocation channel, it sort of sticks, the protein gets stuck. It can't move. So yeah, I mean, luckily we chose the right substitution. Just about fills the space and just about prevents that conformational rearrangement. Yeah, exactly. So it just sort of prevents, we think, we don't know for sure because we've only got the one conformational state for UAPA. And obviously we're working with other sets of mutants and other tools to try and obtain the outward facing conformation, which would give us much more information about that particular region of the protein and how that fits in during the whole transport cycle.
The last, I think, please.
In some cases, the effect of lipids seems to be quite specific on these proteins. Have you an idea if these proteins share some lipid binding moieties, such as crack sequences for cholesterol and so on?
Yeah, that's a really good question. I think the answer is yes, absolutely. So with UAPA, we did, we started off with doing native mass spec. We could see lipids were binding. And then we did lipidomics analysis, which identified PE and PI. Also PC was co-purifying with the transporter. But of course, that doesn't tell you whether it's actually binding at a specific site. And so then, because we have the structure, we're able to do molecular dynamic simulations. And these identified a specific site on the intracellular side of the protein made up of positively charged residues, as you'd expect. So I can show you a picture of it. It's right at the dimer interface. And when we went to do the work on the yeast 4-1 protein, there is no structure of yeast 4-1. So we built a model based on the AT4-1 protein, and I just looked in Pymol for the same motif and found it. Exactly the same kind of thing. I think there were in the case of UAPA, there were 3 arginines. In the case of BOR1, it's 4 arginines, and we weren't quite sure which one was crucial. So we just mutated those, and we can see loss of lipid binding. So yeah, absolutely, I think this is absolutely the case. And this sort of highlights the power of these sort of multi approaches and how powerful molecular dynamics simulations actually is, because you just can't see that in any other way. So an important point also is that we crystallized UAP. That's exactly the same preparation we used for the mass spec analysis. Never saw lipid, never, never, never. So it's either just, it's there, I'm almost certain, because if you take the lipid away, the protein dissociates into monomers. But it's just too flexible. It's just too disordered for us to see in the crystal structure. But the addition of this was just so important for the preparation of this protein. Thank you. No more questions now?
Yeah, I think that there are 2 questions more.
Okay.
Bernadette, can we agree on the fact that when you screen for lipids and you're adding lipids, you are basically adding lipids for stabilization of the protein, which is good for crystallization, but these lipids may not be relevant for the functional control of the protein, while when you delipidate the protein and you still find lipids inside, then you can say that these are functional. Is it possible to draw this difference, or in your opinion not?
Yeah, again, that's another super question, actually. I think the lipids that are most important for function and are going to have the most effect on stability is difficult. I think you're right, Anna. It's difficult to make that distinction. But the ones that are most important for function are going to be the most closely associated ones. And of course, the more closely associated they are, for example, through charge-charge interactions, the less likely they are to be stripped off. But having said that, different proteins, as was discussed for Alexis, is still protein specificity. Every protein is different. And so some proteins are going to bind specific lipids much weaker than others, and the detergent is going to play a crucial role as well. Some detergents we know are much more effective at stripping off lipids than others. But yeah, I think it's difficult to be absolutely clear. If you read some of Carol Robinson's papers on this annulus versus, so the annular lipids versus the specific lipids, I mean, clearly both sets of these have important roles in stability of a given protein, and detergents will strip away almost all of the annulus, and hopefully you'll have some specific lipid stuff.
Daniele, last question.
Yeah. Oh, absolutely. Yeah. So, I mean, molecular dynamics simulations, it's taken me a little bit of a while. I'd be curious to hear about Brian's take on molecular dynamics simulations, maybe Anna's as well. It took me a little bit of time to sort of get into that, but I'm completely convinced this is such a super complementary technique to the experimental work that we do, because we really can't answer all of the questions with experimental methods at the moment, and this just acts as a fantastic resource for being able to see how a protein might behave under specific circumstances in a membrane environment.
Okay, so I think that we can close this session. I would like to thank all the speakers. So thank you again. And the coffee break is in the room here, close, and we start again in 15 minutes. Okay.
Sei solo tu.
Ah, quindi devo
One man
Oh.
session. Alexis, I have a question for you here. Thank you so much. Okay, now then. Tutto il tempo che vuoi. Almeno questo vantaggio.
No, ho aggiunto pochissime slide. Visto che tu
Ok.
avevi annunciato questa cosa.
Me l'hanno detto ieri.
Sì, ma non voglio esagerare.
Però se Ok, allora adesso ti presenta lui, poi vediamo un attimo dove è la tua presentazione.
È nella cartella.
Facciamo così.
Siedi.
Ok, questa la chiudiamo. E quella è la tua? Eccola, quella che c'è scritto in Diveri, giustamente. E quindi poi, tanto lo sai fare meglio di me, fai share screen e poi quando vai qui gli dici, no, qui adesso non te lo fa, ma poi ti faccio vedere, rimango così. Puoi togliere quella riga di
Everyone first of all, I thank Beatrice for the tremendous effort in organizing this meeting. First one. Microphone, it's okay. So for me, at least, is the first in vivo meeting since a couple of years. So thank you, Beatrice. And at the same time, I must apologize for this session will be shorter than expected due to the sudden and last moment abandoned by two of the speakers of this session. So we can take it calmly, and I introduce Cesare Indiveri from Calabria University, who will talk about membrane transporters and novel targets in drug discovery. So I can leave you the floor, and please go on. We have plenty of time.
Okay, I remove my mask. So thank you, Beatrice. Thank you, Alberto. And as Alberto told, this is also on my side the first meeting which is not online. So we wish that this could be the first of a long series, and also we wish for this bad time of Russian war that this meeting could be wish is also for stopping this. Okay. It is now well recognized that metabolism in every organism and also in humans is not only a job made by enzymes, but needs a lot of membrane transporters because the flux of nutrients and metabolites is essential for completion of metabolic pathways. So we see a lot of membrane transporters on the plasma membrane, which allow the traffic of nutrients, ions, and so on, and the membrane transporters also on the membranes of intracellular organelles, such as mitochondria, lysosome, endoplasmic reticulum, and so on. So there are a lot of these membrane transporters, of which you see only some of them in this slide, which are mainly involved in the transport of amino acids, glucose, but there are a lot of other transporters which are involved in transport of many other molecules. In total, in humans, the transporters belonging to this large superfamily, which is called solute carrier, SLC, counts more than 500 members. So this means that the job of membrane transporters is really huge. And about half of them are little known or unknown. So there are a lot of transporters of which we know the genes, but they are orphan. We do not know what they transport. So I will focus on one of them, which can be considered as a benchmark. In that this is LAT1, a membrane transporter for amino acids. It is very interesting because its structure is made by an heterodimer, as you can see in the slide. An heterodimer that contains a protein which is the transmembrane domain, mostly embedded into the membrane, that is the LAT1 member, belongs to the SLC7 family of the solute carriers. And then there is another protein which interacts with LAT1 that is called Cd98 and belongs to a different family, which is SLC2. This protein has only one transmembrane domain. As you can see, most of the protein is faces towards the external side of the cell and contains also carbohydrate moieties, which are absent on the LAT1 moiety. The two proteins are linked by a disulfide that makes them strictly connected. Very, very interestingly, the structure of this transporter is very similar to the structure of ACE2 that everybody knows that is the receptor for the SARS-CoV-2 virus. ACE2 has likely LAT1 an interaction with a transmembrane protein, which is also a transporter. It belongs to a different family, but as you can see, the overall structure is very similar. Thus, information obtained on the LAT1 complex may be also useful for structure-function relationships of ACE2 that may be also useful for fighting against virus. Curiously, the group that solved the structure of LAT1 in complex with Cd98 is a Chinese group, Yan, is the same that solved the structure of ACE2 in complex with BO81. This is the name of the membrane transporter. So we dedicated some years in the study of LAT1 using different models, which are in vitro models, cell models, in vivo models, and also in silico procedure for obtaining information, basic information, on which is the subunit responsible for transport of the two I have shown, which is the substrate specificity of the transporter, and which are the amino acids crucial for the transport function. We have published some papers during these studies that, of course, I will not deal with in detail, but I will give you just some basic information for defining the main properties. First of all, we have extracted the protein from cells. This is a cell line, a cancer cell line, which harbor a large amount of LAT1, expressed LAT1. So it is useful for studying this transporter. And we have extracted the transporter under oxidizing conditions in order to leave the disulfide and the complex strictly connected, or we have also extracted the protein in the presence of a reducing agent in order to remove the disulfide bridge and separate the two proteins. The two complexes, the connected and the separated one, were then inserted into artificial vesicle for studying transport, following the flux of radio-labeled amino acids. And as you can see, in the case of the oxidized form of the complex, we can measure uptake of radioactivity. But the same occurs when the two proteins are not more connected. It means that probably only one of them may be responsible for transport. To define definitely this issue, we have expressed into bacteria the human LAT1 alone, or the human Cd98 alone, and inserted them into different population of vesicles and measured transport. As you can see, only LAT1 moiety is able to catalyze transport, while Cd98 not. Therefore, the conclusion is that LAT1 is the sole competent transport unit. Then we have defined the specificity, which was about the same studied before, but by the first group which approached LAT1. It transports essential amino acids, but the novel finding was that the transporter is asymmetrical. It can transport essential amino acids from outside the cell to inside, but only some of them, mainly histidine, from inside to outside. This novel property was, of course, questioned at the beginning because not all scientists approaching LAT1 were convinced of this result. We had a novel study in collaboration with the University of Vienna after creating the knockout mouse for LAT1. You can see that while a normal mouse can run into the cage, the LAT1 knockout is much more slow in movement. This is typical of a disorder that mimics the autism spectrum disorder. Indeed, a lot of patients around the world carries some mutations which are mutations of LAT1 which are related to spectrum autism disorder that were analyzed in the same study. This is due to the presence of LAT1 into the blood-brain barrier. If the transporter is mutated or knocked down, essential amino acids cannot be imported into the brain, leading to the pathology. As histidine is the major substrate, which is excluded because the transporter has an antiport mechanism, as it came evident in the case of the knockout, in which we found a very large accumulation of histidine due to the absence of LAT1 function. So everybody was convinced now that histidine is the counter substrate for this transport. We have performed mutations in order to understand which amino acid residues could be important for transport. And I will not say the criteria to choose this mutation, but you can imagine that we did a lot of computational analysis before, and we see that there is a specific amino acid, which is phenylalanine 252, that is the only one that, if removed, abolishes the transport capacity of the protein, while other mutations had not a strong effect on the protein. But when we did the kinetic experiments, we also see that there is a strong impairment of affinity of substrate also in the presence of these mutations. Therefore, we concluded that there are at least 4 amino acids which are important for the LAT1 function, that in the outward open conformation of the transporter, that is the transporter open towards the external face of the cell, this conformation of the amino acids allow histidine to be imported into the active site, while in the outward closed conformation, you see that histidine has entered. There is some conformational changes interesting each of the amino acids, but the most striking variation is that of phenylalanine 252 that moves from an open form to a conformation that closes the cavity, thus behaving as the gate of the transport. Now, the question is LAT1 druggable? The response is yes, but we have to think about the role of LAT1 in the human organism. The protein is expressed, I have told before, essentially in the blood-brain barrier. Other researchers found that it is also expressed in the placenta barrier, while it is not expressed or expressed at a very low level in all the other tissues, except when a tissue became cancerous due to transformation. In this case, the expression of LAT1 increases a lot. Even tissues that normally do not express at all LAT1 have a very high expression of LAT1 when they are transformed. This is due to the need of this tissue, of course, of a large amount of essential amino acids for which LAT1 is important. Thus, as you can imagine, it is very interesting to find inhibitors of LAT1 for chemically knocking out the transporter, hoping to have a further tool for anti-cancer therapy. So we have used 2 different approaches for finding inhibitors. That is the competitive inhibitor approach, that is finding molecules similar to the substrate that may behave as competitive. The procedure that we have used for finding such molecules is based on a computational analysis before for finding molecules that may interact with the protein, screening virtual libraries. then performing docking analysis and finding some lead compounds which may be predicted to interact with the protein. We have found, interestingly, that only molecules which share an amino acid function, that means a carboxyl group and an amino group, can interact actually with LAT1. Indeed, docking analysis gave normally a lot of compounds, also not sharing these functional groups. But when we did the validation by in vitro or in vivo, not in vivo, in cell, ex vivo analysis, we found that only those compounds which have a carboxyl group and an amino group can inhibit the transport. This is a paper that we did with the University of Vienna, also in this case, but a different group. After a screening of a large amount of compounds, you see that we arrived to very few compounds, 13 compounds that were able to inhibit in some way the protein, but only 2 of them had an EC50 which was in some way interesting around the micro. Of course, we are not the sole group working on LAT1. A lot of other groups in the wild were looking for inhibitors, and so far the most active inhibitor for LAT1 is this compound called JPH203 that has an EC50 lower than 0.1 micromolar, and it is actually in clinical trial, even though its effectiveness towards against prostate cancer is still not well defined. The other approach that we used is the covalent inhibition approach that, of course, cannot be based at the first on computational analysis, but on pharmaceutical chemistry, and we identify a compound that is a thiazole ring that has the ability to interact with cysteine residues. Why we sought this compound? Because LAT1 has 2 cysteine residues in the active site, as I have shown in the slide before. This thiazole ring can be opened in the presence of a cysteine residues, forming a disulfide or a trisulfide. That is a stable linkage. Thus, if it would work as an inhibitor, it should be a much more stable inhibitor. So, together with 2 groups of pharmaceutical chemistry, they synthesized a lot of compounds, of course. And as you can see, more than 80 compounds were synthesized and then checked for inhibition on LAT1. And also in this case, that is not a computational analysis, but a lot of experimental work, we got only 2 compounds with an IC50 in the range of 1 micromolar. These 2 compounds, in particular, we tested one of them, were able, this is a computational analysis of the possible interaction that predicted the compound to react with cysteine 407, but when we analyzed the mutant in which cysteine 407 was substituted, the EC50 was much higher, demonstrating that indeed the compound was effective because it reacts with the cysteine residue. Analyzing the effect on cells, you can see that giving the compound at concentration of 10 or 100 micromolar to a cell population, still see a cell that contains a lot of LAT1, expressed LAT1, you see that the number of cells is much lower after treatment with the compound. After 24 hours, after 48 hours with respect to the control, but more interestingly, it was still active also after washing. You can see that after washing, there are still very few cells compared to no inhibition. This means that indeed the compound acts by a covalent linkage. A control was performed with one of those compounds which had a much higher EC50, much lower affinity, and as you can see in this case, there was no inhibition of cell growth. Now, do we have further perspectives of improving the chemical knocking out of this transport? This is an important issue because competitive inhibitors are, especially if they have a very low EC50, they have a strong inhibition effect. But, however, also by clinical trial, there is still not a complete blocking of cancer progression. So it will be interesting improving, still improving the chemical knocking out. So we sought to move our attention towards the Cd98 protein, which, as I told before, interacts with LAT1. Cd98 is a glycoprotein, thus it's very likely that it will have some function in trafficking of itself and probably also of LAT1. Thus, to understand this, we have mutated the protein, removing the glycosylated moieties. And as you can see, when we performed 4 mutants of asparagine residues, removing all the sites that can be glycosylated, the molecular mass of the protein is lowered from that of the wild type to a molecular mass of the quadruple mutant that is coincident with the sole protein moiety. All these other proteins that you see, which have been obtained by Western blotting, are the single mutants, the double mutant, the triple mutant, and you see that the molecular mass is always decreasing. The experiment that then has been performed is an experiment in which we measure traffic by using a strategy that is based on Brefeldin addition. Brefeldin is an inhibitor, a general inhibitor of trafficking. This allows us to start to define a time zero of trafficking. So time zero is the time in which Brefeldin is removed and the protein can start its trafficking towards the plasma membrane. As you can see, in the case of the wild type, the protein is at time zero all inside the cell. After 1 hour, most of the protein has reached the plasma membrane. After 3 hours, all Cd98 is on the plasma membrane. While in the case of the quadruple mutant, time zero, of course, is the same as wild type, but also after 1 hour and after 3 hours, the protein is into the cytosol. So trafficking is abolished. So it was very interesting to see if also LAT1 is interested by the same phenomenon, because being LAT1 interacting with Cd98, removing the glycosylation moieties, probably also LAT1 remain inside the cells. Indeed, we found that while in the case of wild type Cd98, of course, Cd98 goes to the membrane, but also LAT1 that is in this slide represented by a different color, also LAT1 goes to the membrane. Merge here show you that the two colors are exactly coincident. In the case of quadruple mutant, Cd98 remains into the cytosol, and very interestingly, also LAT1 remains into the cytosol. So the trafficking of LAT1 requires Cd98. That means that a possible intervention on Cd98 may reinforce the inhibition of LAT2. This, of course, should be done in the future. These data are part of a paper that has been submitted yesterday. So there is still a lot of work to find possible inhibitors, specific inhibitors of Cd98 traffic. Just to show you that LAT1, as I used as a benchmark, is not the sole protein interesting for drug targeting and drug discovery. Here there is a list of other transporters which are involved in drug targeting. In particular, there are two transporters, SLC6A4 and 5A1, which share inhibitors that are already used as drugs, in commercial drugs, that are antidepressant or anti-diabetic drugs. Anyway, these drugs were not designed on the transporter, are drugs which were used also before, and then after the well-knowledge of this transporter, it was discovered that they indeed interact with the transporter. Then there are a lot of other transporters of various families which are involved in pathology, essentially cancer, and for which several researchers are finding inhibitors, as I have told in the case of LAT1. Then there is this SLC-A19, which is the transporter linked to ACE2 that may be a target for COVID-19 and is already a target for diabetes, and so on. Now there is a last minute issue that I have inserted because Beatrice told that there are two speakers not coming. So I have just few slides more that concern this topic. Some of the transporters are not considered drug target just for inhibition, but they are drug targeting that are able to deliver drugs. They are transporter of drugs, because drugs in these cases have similar structure with respect to the substrate. So they are not only inhibitors, but they can also be transporters. So there are a series of transporters in the SLC22 family, which are involved in drug transport and are more so important for this that FDA already considered these transporters as important for designing new drugs. New drugs must take into account that they should be transported by some transport system. So I will give you some information on SLC7A5 LAT1 concerning this novel feature of being important for drug delivery. And we found that a compound that falls into the nutraceutics, there is a class of compound that now has a big evidence and fashion in the scientific community, Allin is a compound that is found in garlic and is believed to have several activities in the human body. Allin, we have found by computational analysis that can interact with the LAT1 binding site, and in particular, it can interact with the phenylalanine 252. This is the map of interaction that shows interactions of allin in comparison with interaction of histidine, and the interactions are mostly the same. So we have tested allin as inhibitor. From this experiment, you realize very easily that it is a competitive inhibitor. This is the control. These two curves are those obtained with different concentrations of allin, but most interesting thing is that when we perform this type of experiment, which we fill the vesicles with the labeled histidine that can come out vesicles, only if we add histidine outside, this is the control, histidine can come out, or when we add allin, that is this curve, histidine can come out. That means that allin, as the physiological substrate histidine, is transported by LAT1, thus can be imported into tissues in which LAT1 is expressed. These are also last results that are on a paper in Frontiers in Pharmacology that is in revision. This is the sea of Calabria, as you can imagine. And this is my relatively small group. There are 4 scientists which have now, 3 of them have a fixed position, 1 is still postdoc, all the others are thesis students. And here is the list of collaborations we had with several others. Thank you very much for attention.
Thank you, Cesare. We enjoyed a lot of this last discussion on relatively little, to me, known transporters. So we have time for some questions. Anna,
very interesting. I have billions of questions. We have billions of minutes, I think. So the first one is just a curiosity. Given the similarity of the ACE2 receptor, is the spike protein recognizing the LAT1 similar or the other one?
No. Spike recognizes only ACE2. What we have found that there is a molecule that interacts with the transporter bound to ACE2, that is nimesulide, that blocks the activity of the transporter bound to ACE2. Nimesulide, we have proposed nimesulide as a possible anti-COVID drug. And there is a group in Russia
there was a group in Russia
that has tested this possibility, and they found that nimesulide is more active with respect to other anti-inflammatory molecules towards COVID-19.
But my question was, does the spike protein recognize which of the dime, which component of the heterodimer?
ACE2 only, not the yes.
The transmembrane part.
This domain of the spike protein can bind on the external side of ACE2, and when we have thought about the inhibitors, but because when the virus binds to ACE2, there is a big conformational change in ACE2 that is transmitted to the transporter part of the complex. So our opinion was that if we block the conformation of the transporter part, probably the binding of the virus could be impaired. This was the principle why we introduced this.
My other question is, given that LAT1 is in on the brain blood barrier,
Yes.
do you believe that it's possible to evolve in vitro the protein until it's able to transport other interesting drugs for entry? They have to be eased in life.
This is a crucial question.
Yeah.
Indeed,
In vitro evolution, probably.
There are some drugs like gabapentin, which are transported by LAT1 into the brain. And there is an active effort by several groups to construct prodrugs made by an amino acid moiety with another part that should be delivered to brain that could be transported by LAT1. This is a very interesting issue.
Okay, thank you. Another one. Oh,
I have another question. Sorry. You were mentioning that the transport protein is LAT1, the transmembrane one. But how did you measure the transport activity if it doesn't go by itself onto the membrane? If it's not trafficking into the plasma membrane?
Measured the transport activity. The final experiment was producing the two proteins separately to bacteria. Then insert the two proteins into different pool of vesicles, and then measuring the. Okay.
Sorry. So as I understand, there is the effect of the CD protein on the specificity or activity is non-existent. No regulation whatsoever.
No, it is involved just on traffic. There are some papers telling that there is some very small change in specificity for some amino acids, but just this that probably may be due to more difficulty to reach the protein when the Cd98 is up to the transporter, but just some few changes in specificity.
But are there any known function for CD98 apart from, I mean,
a lot of function. This is a very interesting topic because it has not only the function of trafficking to LAT1, but it has also the function of cell adhesion. And so it is involved in cancer much more than LAT1. So this is very interesting.
Thank you.
Thank you for your very nice talk, as usual. I would like to know something. Maybe you're not, you haven't worked on crystallography. I know that. But what would be the reason? How did people achieve two different conformations of the, the outward open or outward? How was that achieved? Just by chance, or was the rationale?
Not by chance. You know that transporters have different conformations during the transport cycle. The two different conformations, the outward closed, the outward open, were achieved by binding an inhibitor. The outward close was obtained with an inhibitor bound into the active site. The outward open was obtained with the free protein.
And how about the inward open closed?
No, they are not available. Thank you very much.
They're more expert than I am because I'm very young.
What does this mean?
Digita risposta.
Okay.
Vai. Allora, balance, young team, I need some help. Let's tidy up a
Do
little
you
bit.
know
Mettiamo un po' a posto.
where the restaurant is? Ah, gosh, I was really hoping you would, that we could go together.
Yeah, we can.
Find out from, just wondering about the restaurant. Downloaded the map. So much going on. Yeah, well, because I think it's not far to walk. So,
No, but it's early to go.
oh yeah, no, not now. We go.
No, I mean, point is,
Yes.
yes. Alle 8. And
Yeah.
to the guest house.
Yeah. Yeah, but again,
Yeah,
great.
we
Freezone is
can-
much better than
You can say at what time 7:30 or
fine. Why not? Yeah, in the reception and then we can go.
yeah, okay.
Excellent.
Should I turn it off? I think I should turn it off.
Oh, maybe, yeah.